Hello everyone, I'm Jahan Sandhu, and today I'll be teaching you what a virus is, the different types of viruses, the structure of the types of viruses, and the different methods viruses use for reproduction. So without further ado, let's go. So to begin, we should define what a virus is. A virus is an incredibly small infectious agent that relies on a living host to reproduce. Viruses can be found in almost all forms of life, and their extremely small size is the reason they are able to live inside and infect cells of other organisms. Viruses are considered alive because they lack many of the necessary properties that go hand in hand with life. For one, viruses are not made of living cells, which plants and animals are. Another thing is that they do not react to outside stimuli or try to maintain homeostasis, or balance, in their environment. An example of homeostasis in humans is that we have a specific body temperature that our body tries to keep us at using shivering or sweating. But the main reason they are considered alive is because they require a host to reproduce. All viruses are structured similarly with two main parts. There is the DNA/RNA center, which they use to infect cells and reproduce, and the protein coat, which protects the important DNA or RNA. So next, we move on to the types of viruses. There are two main types in which all viruses can be classified. These are normal viruses, which we have here outlined in blue, and bacteriophages, or just phages, which we have outlined in red here. Normal viruses can infect the cells of living organisms, which range from animals to even plant cells. Bacteriophages get their name from the fact that they infect and replicate within bacteria. As you can see, bacteriophages look significantly different from normal viruses. This is because bacteriophages have evolved to attack bacteria, whereas the regular viruses have not. Not all normal viruses are the same though. In fact, they each have a different size, shape, chemical composition, and structure of their genome. All different species have a unique virus that infects the host and causes different effects. Now I'll be going more in depth on the structure of bacteriophages. The average size of a bacteriophage is about 6.5 nanometers, which is about 1,000,000 times smaller than a millimeter. The phage is a protein coat, also known as a capsid, that surrounds the precious RNA or DNA, never both. There are also times when the virus will enter its infective form. In this form, we have a particle located on the outside of the cell that is called a variant. All variants contain at least one special protein synthesized by specific genes in its nucleic acid. We also sometimes have a lipid and protein structure known as the viral envelope. The viral envelope structure is made from the host cell membranes. The virus also has a couple of leg-like fibers called tails, which allow it to stay firmly secured to the bacteria it is infecting. When the tail is secured to a bacteria, the sheath becomes active and allows the base plate to pierce through the cell membrane. The bacteriophage can then go on to infect its target by injecting the DNA that it has stored in its head. So now we're moving on to the reproduction of viruses. So viruses have two main infection strategies. One of these is the lytic cycle. Viruses that use this strategy are called virulent viruses. In this type of infection, a virus creates many new bacteriophages, but needs to infect a bacteria cell first as a sacrifice. So a virus will latch on to a susceptible cell and insert its DNA. This DNA inside the bacteria then starts to decompose the genetic material of the host. This causes the virus to hijack the ribosomes, which are the protein factories in the cell, so that the new proteins being created will go towards building new copies of the bacteriophages. The pieces of protein gather around leftover nucleic acid from the DNA that was inserted, and are assembled into new copies of bacteriophages. The cell is eventually so filled with viruses that it tears open and the viruses are set free. These new viruses then go on to find new bacteria and continue the lytic cycle. The second main type of infection strategy that viruses will try and use is called the lysogenic cycle. Viruses that use the lysogenic cycle are known as temperate viruses. So this strategy is when the virus replicates inside of a cell without destroying the actual cell itself. This is the main difference between the lysogenic cycle and the lytic cycle. But another big difference is that it takes much longer for this cycle to occur than for the lytic cycle to occur. The first two steps of this cycle are pretty similar to the lytic cycle. A bacteriophage latches on and inserts its DNA into bacteria. The difference starts here. Excuse me. When the viral DNA is injected, it integrates into the bacterial chromosomes in order to become a prophage. A prophage is just the genetic material of the bacterial phage incorporated into the genome of bacterium. It's able to produce phages that activate in a certain way. Once the bacteria starts to undergo binary fission or splitting itself and creating a copy, the prophage is copied along with it and becomes part of the daughter cells. Stealthy, right? And this way it can keep traveling along. As you may have guessed, the daughter cells will continue to undergo binary fission with the copied prophage still inside of them. This means that the copied virus will stay inside the cells and keep multiplying along with them. Now at this point, the prophage can keep being reproduced or exit the cell and begin the lytic cycle. In this way, it's possible to be infected but not notice it for quite some time. So to recap, the lytic cycle is fast-acting but kills the host cells very quickly, and the lysogenic cycle can take a long time to activate, but will go unnoticed for just as long. Thank you for watching my video about viruses. I hope you learned something new. Take care.